Thank you so much, George, for this privilege. So I graduated from Argentina in 2000, and in 2001 I joined the José Romagnoli group in Sydney, Australia. Then I graduated from his PSC online group in 2005. And at that point in time, I went to Europe to do my postgraduate, essentially reporting to Kostas Pantelides. It was also that time that I had the privilege to be influenced by the academic mind and leadership style of Stratos Psikopoulou and Wolfgang Marquardt, who were also in the research network I was participating. Around 2008, I joined PSC Limited, was involved in the development of GPROMs, consulted for several Fortune 500 companies, until in 2006, I decided to come closer to George, across the Atlantic Ocean and joined Amgen. My story, my personal connection to George is a little bit different to most of you. I got to meet George in 1999, and that was through his book, Chemical Process Control, Introduction to Theory and Principles, which was the undergraduate book that I used to learn about this fundamental aspect of who we are as chemical engineers. And I say that I got to meet George then because it was through that book that you can say that his persona and personality certainly transpires as you read it. So it wasn't until 2009, so essentially 10 years later, that I actually met George in person for the first time. There was a PSC Escape conference in Salvador, Brazil. And what struck me at first was his keenness to be interested and to be helpful to the people who were around him. And I think we can say that George is definitely one of the greatest in our field, because precisely he's touched many of our lives in various different ways. When I had the opportunity to move to Boston, I said this is a great chance to connect closer with George, and unfortunately I didn't realize on that opportunity because things got easier. And now I learned that George first took a sabbatical and now he's about to retire. But hopefully that will mean he will have a little bit more time available, and perhaps he can take one of his newer students of his in more of a mentorship role perhaps. So having said that, it's time to introduce our first speaker, the person who certainly does not need an introduction. This is Rex Rekiaitis. George knew Rex several years before he actually met him through his research work with Doug Wild at Stanford. He became interested in his research in numerical optimization, particularly on geometric programming and linear uncertainty. Because of their overlapping interest in process systems engineering, Rex and George have enjoyed a very close relationship over the last 40 years. Now, in terms of his education, Rex got his Bachelor's of Science from the Illinois Institute of Technology with highest distinction in chemical engineering, and then his Master's and PhD from Stanford University, also in chemical engineering. He's been at Purdue University since 1970. And today he's the Edward W. Cummings Professor of Chemical Engineering. He's the Deputy Director of NSF Engineering Research Center for Structural Organic Particulate Systems, and co-director of the Pharmaceutical Technology and Education Center. Among his many distinctions is the U.S. National Academy of Engineering in 2007, various awards from the American Society for Engineering Education in 1991 and 1994, and finally the ACHE Cassivision 1984 Award for Computing and Chemical Engineering. Today, Rex is going to talk to us about prospects and challenges for PSC in health.
For that nice introduction, and certainly thank you, George and Richard and Paula, for the opportunity to join this historical event to celebrate a birthday and a transition. The connection certainly between us has been quite long. My first recollection of you was really when I was an assistant professor and you visited the campus and walked into my office and began to speak Greek to me. And I had to acknowledge that it was Greek, and that my descent is really not Greek, but Lithuanian. But, then one could talk about the affiliation between these language groups and so forth. Since then, we've certainly had a number of interactions. In terms of family tree, I have to join Tom, who was here earlier. My academic grandfather was Andy Akravos, so there is this parallel academic tree, but I'm sure it's equally as distinguished. So, my job here is to tell you a little bit about the potential role for PSC in healthcare. And so I'm going to take a little bit of a different slice than some of the previous speakers who focused more on technology. I'm really focusing on what are the opportunities in this domain. And as a result of doing this kind of a different slice, you will find that I intersect quite a bit with some of the previous conversations that Ka had with you, that Richard just had this morning, and so forth. But that's okay. At our age, a little bit of repetition is a good thing because the cells don't absorb stuff as readily. So what I'm going to do is give you a very brief current context, and then I'm really going to talk about some challenges in four areas. Four areas. The design area, the manufacturing area, and then a couple of less traditional areas that we can pursue. I'm going to present these areas sort of in terms of a challenge a goal, and then look at these goals and see, well, what are the barriers and where are we right now that gives us hope that we'll be able to address those challenges in the future. Now, as you recall, early on in this seminar, there was some reference to George wanting to see the money. And so the first thing I was going to do is indeed show you some big dollar figures just to motivate you properly. The first of these dollar figures is just to impose on you the fact that healthcare really is a pretty substantial business. It's almost $8 trillion worldwide expenditures. And as usual, the US is quite efficient in spending lots of money, and not as efficiently as the rest of the world. As you see, it's about 18% of US GDP. So it's really a rather massive enterprise. And if you look at how that dollar is spent in the US, really, by far the biggest is hospital care. The next is professional services. That's our friends, the doctors and dentists and all these wonderful people. And then prescription drugs is really the next largest. The other parts are really essentially longer-term care, personal care, sort of the overhead of managing a healthcare system. But it's a rather dramatic amount of money. And traditionally, the PSC and the chemical engineering community really have closest affinity to the prescription part, because after all, a good bit of that is chemistry and biology. However, I think some of the opportunities now are really getting closer to bridge over from the drug to the professional services and make that sector more efficient and more productive. So those are a couple of the things that I will want to talk about in this presentation. Now, just in terms of a quick briefing for those of you who are, of course, young and don't care about this area, but, some of us do worry about it a little bit more because we want to rely on these products. So in the US, I should say, in the world, the pharma industry is a $1 trillion business, and it is truly a global business. But it's a very heterogeneous business. If you look at the market, there's relatively slow growth in terms of sales in the developed world, except for the US, largely because of budget constraints and really stagnant population growth. In the US, it's kind of a bifurcation between more and more prescriptions being generic, like 90% or thereabouts of prescriptions written are really generic drugs. And then you have the other sector, which is the specialty sector that's getting a lot of attention in the press. We're talking about treatments that are tens of thousands, hundreds of thousands of dollars. And that's the market that probably the kind of technology that Richard was talking about addresses. But we forget sometimes there is a large volume of products that are really generic. They're not innovations, one just has to make them better. There is also another sector that is sometimes in the literature called the farm emerging sector. So these are countries in which there is a developing middle class. And as the country develops, you progress from worrying about acute conditions to actually treating chronic conditions. The chronic conditions generate steady cash flow. From a business point of view, you want to pay attention to them. In the rest of the developed world, it is really about treating acute conditions. And again, technologies such as the flexible technology Richard was talking about really can address that. So I want you to appreciate it's a very heterogeneous mix. And they're really not a solution that's appropriate for all of these sectors. Overall, we all know it's a high-risk, high-payoff enterprise to put a new treatment into the market. It is highly regulated, both by the European Medical Agency, the FDA, and so on around the world. In most of the world, prices are controlled. In the US, it's becoming to be more controlled. And probably will continue to see price controls. In terms of cost and quality, really it's the generics that have the largest percentage of the sales dollar tied up in manufacture. The innovators less so, but our wise people in organizations like McKinsey, I don't know where they get their wisdom, they claim those costs could be reduced by 50%. But there really is a sector here, the specialty sector, where manufacturing cost really does not figure in. And the incentive for innovation in the specialty sector is really to get a new innovative drug out faster. It's not about manufacturing costs at all. All right, the other interesting feature of the industry, increasingly the industry is going to outsourcing not just for capacity, but also for capability. And that's kind of a curious phenomenon because capacity outsourcing has always been in place because it's so hard to predict the market for a new product. So you may underestimate the capacity you have, but this capability part is a little bit troubling. So just as a brief tutorial, I'll remind you that the life cycle of a drug goes from discovery all the way through design of processes and products, trials, regulatory approval, out into the market, into the supply chain. When it's in the field, its performance has to be continually monitored and improved. And eventually, you reach patent life, and at that point, the generic takes over, and those generics are traditionally been smaller molecule organics, but increasingly now is the emergence of biosimilars. The boxes in black, or in dark, are really the areas where clearly chemical engineering and particular process systems engineering have major impact. We've already talked about design of processes for the active. We talked about product design. Manufacturing is clearly in there. Probably not as much discussion of how to participate in clinical trials or how to actually enhance the use of the drug out into the field. And I'm going to talk about those a little bit. Now, the pharmaceutical development, just again as a primer for those of you that are not into this area, we always talk about drug substance, or API, and roughly can be classified into small molecule, large molecule, so synthetic, organic versus biologic. And then we have a number of product forms, from solid oral down to transdermal, and lots of innovative technologies that have yet not had a big impact on the field. Clearly, over or nearly 2/3 of delivered drugs are really in oral form. Injection infusion is the next part, and then the rest really are smaller parts of the market. So from a point of view of manufacture, it's the first two are probably the ones that deserve the most attention in terms of volume, although others certainly have interesting features to them. Now, in all cases, we're always going from a molecule to synthesizing a process for this drug substance, to designing a product that will deliver that substance in such a way that there are desirable patient outcomes. And as Ka and others have pointed out earlier, while we don't worry about the discovery part of the molecule, these last three boxes really need to be dealt with in an integrated fashion, because ultimately the benefits is only achieved when the patient reaches the desired therapeutic state. So clearly the first problem then is a process synthesis problem. And the only complication here is that the bulk of the industry is really batch, and I would expect that the generic sector will stay batch for a long time to come. So what are some of the challenges in these areas? Well, it really, particularly given how important good processes are to generics, the goal would be that by 2040, we would have industry use of knowledge-based and model-based tools in process synthesis. There are barriers to that. The complexity of the chemistry is one. Probably the key feature of pharma is the focus on impurities and reduction of impurities, because that entails a significant cost. You have to identify toxicities and all that sort of form. Estimation of physical properties is really a key problem, because certainly on the innovator side, it's always a new molecule, so there is no history. However, there are strengths. I think one of the attractive developments is the industry has now begun very actively to talk about models and the use of models. Also, if you look at most of the development groups, they're now well infiltrated by chemical engineers who can bring that kind of skill set. Before, it wasn't all that long ago when I would visit Lilly's development team and there wasn't an engineer to be seen. But now, PhD chemical engineers with solid traditional backgrounds in reaction engineering and separations are present. So there is an audience for it. We now have fairly extensive libraries and chemical reaction databases and equivalent knowledge building on the biological side. And of course, George and his group have done quite a bit of work in the process synthesis area that was really very pioneering, and in fact, I would say ahead of its time. I recall, or you may recall, there was some early work on design kit, followed on by some very interesting work on batch process synthesis that Andreas Lininger and others contributed to, which really put together a framework to really do the reaction path planning, the refining of the unit ops, in terms of even down to facility allocation and analysis. And really quite an interesting development, addressing both sort of the structured approach to building these processes, as well as the ability to study a particular operation in fairly great detail and present alternative process views that could be used as a communication vehicle. Of course, it wouldn't have been George's group if it hadn't been infiltrated by some AI-looking stuff like means ends analysis and non-monotone planning and so forth. This was commercialized. I believe it was much ahead of its time. I think if one could wish for an alternative history, I would say this would be the time to begin introducing this technology, because the industry has infrastructure. They have people who can actually use and understand these tools. So it's nice to be a pioneer, but it's even nicer to be there at the right time. But perhaps Richard will help make that happen. On the product design side, we've already discussed products pretty extensively. It's pretty clear that most pharma products are formulations. And so they really have the usual component selection, the active, a number of non-therapeutic components that have different roles, from stabilizing the active to facilitating manufacture to facilitating delivery and actually to protecting the product. However, as we said, there is really significant concerns with product performance, and what you do in manufacturing, the process conditions can affect its effectiveness in delivery. The patient response is really quite important, while, as we talked earlier, to actually factor in the therapeutic response is difficult, but to factor in the pharmacokinetic effects, one can do that. And on top of that, of course, there's also the issue of stability. So for this category of product, shelf life is very, very important. Okay. So that makes the product design problem a challenging problem. Now, just by way of an example, take the simple tablet. Really, you have the API. Suppose it's a blood pressure regulator. What you'd like to do is just to dump that molecule right where it's needed and eliminate the complexity. But as you now know, that's not feasible. So what we have to do is we have to configure it into a tablet. And in order to do that, we have to worry about the crystal properties, the polymorphic form, the crystal size distribution, the right excipients, produce the granules total, form a tablet, coded, and then, of course, it would go to the patient. So what do we need? First of all, this patiently assembled tablet now needs to do the opposite, needs to fall apart. It needs to release the active ingredient into the bloodstream, and then, of course, it needs to do its good. Once you get to this stage, of course, this part is chemistry, right? So there's not much we can do in terms of product design and doing this excipient selection. That all has an impact up to here. So that's why I say, really, for product design, probably the pharmacodynamic part isn't as essential as it might appear. Now, the same sort of issues, however, arise in the other product categories. An injectable product, again, needs to be formulated in such a way that it is stable, that it can be delivered in significantly in concentrations that allow a dosage to be delivered in 1 to 5 milliliters. And so there is significant formulation problems in the other product carriers as well. So from a product point of view, it would be a goal, perhaps a stretch goal, to have the industry-wide use of knowledge and model-based tools for product design. I think there's been significant progress made already. However, there's barriers. Formulations are complex. Currently, they rely on human sort of internalized knowledge. Estimation of properties, even of powder blends, is difficult. Prediction in performance in vivo, for example, the absorption part, is still not perfect. However, again, we have development groups that have significant number of chemical engineers. There's been lots of work on product design frameworks. Ka gave us an excellent exposition in that area. There's been significant advances in the modeling on the patient side, particularly models of what happens in the GI tract, PBK models, transport models, and so forth. Now, George has not directly worked in this area, but certainly Joe Back, whom we heard earlier in the week, started the chain by looking at molecule design, and I'm happy to see that he's continued that thought process into product design, because it's very much needed. Okay, the third area, which is very traditional chemical engineering, of course, is that of manufacturing. And as I mentioned earlier, the current state is batch operations dominate. Certainly in the generic sector, it will continue to dominate. I'm convinced of that. There's been a lot of attention to continuous manufacturing, particularly on the product side. Certainly significant attention now to small molecule and progress being made in biologics. The emerging areas are topics that Richard addressed, sort of point-of-use manufacturing, mini manufacturing systems that bring the product closer to the patient. How far that will progress, I'm not sure. I suspect it will be more likely for the high-end products. Now, traditional batch world is usually described by cartoons such as this, where one takes what happens at the bench scale and sort of linearly scales that up to the manufacturing scale. It just makes the vessels bigger. That's probably not a fair characterization of the industry, but it's a cartoon. So the old style design is one where work is done by process chemists, engineers are given some scope for improvement, the package is transferred to manufacturing, scale up occurs by trial and error, and then, of course, continuous improvement is limited simply because of the requirements to get approval for changes from the FDA. So that creates a framework of traditional manufacturing that makes innovation difficult. However, I think we can certainly do a lot. There's been, my idea would be to adapt as goal, even for the generics by 2040, to have really integrated supervisory control and scheduling methods in place. These plants are all multi-purpose plants, so scheduling of product campaigns is very important. The barriers are there. There is still the legacy of quality by inspection rather than by controlled manufacture. There is still a legacy of limited measurement and automation, lack of trained personnel, and the perception that dynamic models are complex. Strengths, we've got chemies infiltrating more and more. The PSC community has done quite a bit of work in this area. As you saw from Kostas's presentation and the Aspen presentation, we have lots of good tools available. And of course, George and his group have done significant work in this area, beginning with sort of the work on operating procedure synthesis. On the continuous side, our community is well aware of the hierarchical levels that Richard summarized, from monitoring to real-time process optimization. We can take that technology and move it over to things like tableting lines, where even though these processes are not that complex, what makes them complex is the fact that in order to measure things online, we have to use spectroscopic methods and other more expensive analytical tools. There are also some constraints on what we can do. The FDA, of course, is concerned about product quality, so this aspect of non-conforming materials tracking that Richard referred to is really a very key issue. That means you have to have a sensor network that can detect that. You need to have fault diagnosis technology that allows you to identify when this is happening. And fortunately, this community has worked hard in this area. And so we can set as goal, I think, adoption of continuous manufacturing across the industry for those products and recipes that justify that. Clearly, some of the barriers are there's lots of iron in the ground. So how do you convince a company to invest in new things? There is still kind of a lack of clarity on the business case for when to go to continuous and when to stay with batch. I think that will get gradually resolved. Certainly, there's slow progress on the part of vendors. There's still a lack of trained personnel to really do this kind of stuff, but certainly our community can help address that problem. On the positive side, the agencies themselves are very much encouraging innovation in this area. Continuous is very much discussed around the industry, even in the trade journals. We have some of the tools in this community that can help take our TPM technology and use it in that new sector. And of course, George's group has done a remarkable body of work in monitoring and data reconciliation trend. I mean, all of these topics that really help support that triangle have been developed in his group. So I think that's very exciting, and I do believe this is among the goals that actually can be comfortably realized. Now, the last two things I want to talk about is, first of all, the product pipeline management part. Just very briefly, this is a workflow that goes from discovery through process design, product design, clinical trials, development and manufacturing facilities, and out the door. This is an interesting decision problem. One needs to, out of discovery, select the products that should be advanced. One needs to assign resources. One has to manage the clinical trials and produce clinical trial quantities as you go along. You then have to plan the production facility and, of course, respond to failures, because as a product fails, you reassign resources. So this is the kind of a decision problem that our community has worked with. There are some interesting complicating issues. One of these is more and more the industry is going to in-licensing, so injecting product ideas into this workflow when the internally generated products aren't sufficient. There are other issues that increasingly become interesting now. More and more talk about adaptive clinical trials. So that means the clinical trial quantities will vary, and so how do you supply that in a cost-effective way? And of course, you want to manage this across the whole product portfolio. So this is, an interesting and challenging area. I believe that there is scope for the industry to adopt, quantitative type tools to support these decisions. Unfortunately, there's a limited history of using model-based tools. Part of the requirement is really integrated information systems, and those are evolving. A lot of the industry is outsourcing clinical trial management, but typically those folks really came from those companies anyway, so they really don't have any new skills that they didn't have. And of course, from a PSC point of view, these are inherently multi-stage stochastic programming problems. So that makes life a little bit interesting. However, I think the industry realizes that integrated information is necessary to support development. They know costs are escalating. We're talking about $2 billion plus per successful drug. The transition to adaptive will require some flexibility in supply chain. And of course, our community has begun to do research, and I just mentioned some work of Ignacio and Selin and others that have begun to look at this issue. So my assessment is that this is an area where we really can contribute. That crosses over from purely manufacturing. The last thing I wanted to say a few things about is the personalized medicine area. And my take on personalized medicine is not use of genomic information to select the treatment option, but rather to worry about the proper dosing of the patient. And just some statistics, 50% of the population in the US takes medication. Elderly on this slide refers to those 70 years old and older, so George, as of today, you qualify. There's a growing prevalence of drugs with narrow therapeutic index, so getting within that window is becoming challenging. And there's plenty of statistics that show adverse drug events are really a significant drain on resources and people. And the goal of preventing adverse drug events has really been outlined in a number of public government type documents. So what is the concept? Well, I think the work that Frank Doyle and colleagues have done in the artificial pancreas is in some sense the idealized individualized treatment, where you have an online monitor to monitor glucose. You have a reservoir and a pump. You have an MPC or simpler control algorithm that simply closes the loop and doses the patient appropriately. What makes this work is really the sensor. That's really the key component. Our PSC community can develop the models, but the sensor, the real online sensor and analysis is really the key. So in this area, the more likely scenario for most drugs is a physician in the loop model, where you use population models, sort of in Michael's terminology, you build a population prior, You then collect information on the specific patient. You then use Bayesian methods to come up with a posterior, which is designed for that patient. And then you use optimization methods to come up with the dosage. So that's a reasonable model for this kind of approach for many drugs. And there's been some work done in this area by various contributors. The other part of it is actually developing the dosage form. The easiest way to individualize the dose is, of course, by injection or infusion. But from a patient point of view, the tablet or capsule is the simplest way to deliver it. And we've done some work in this area to build very simple devices that reliably produce tablets or capsules or film deposits using various formulations from solutions to suspension. So just to summarize that, the goal here will be to have individualized medicine adopted in most, particularly pediatric and oncology. Those are the two areas where dosing is very critical, particularly pediatrics. We're currently working with pediatricians, and I'm amazed how they manage to treat patients. The barriers are, of course, the measurement part. You want to sample a small amount of blood. You want to do a rapid analytical method so you can make a decision. The computations, as Michael pointed out, can be hairy. But part of it can be done offline, part of it can be done online. Model development, probably one of the most expensive things we've seen so far in our work is really the clinical studies. Strengths, the clinicians very much realize they have a problem, particularly pediatricians. There is access to e-medical records, so you can pull up patient information. The clinicians are beginning to use advisory systems. They're very primitive now. They simply list what was dosed in the last several successive cases. We've demonstrated the ability to do this with retrospective data, and there's still some work that needs to be done in flexible dosing, but I think we're heading in that direction. So let me kind of conclude with a summary. First of all, I believe healthcare sector is indeed very fertile for PSE. I think it's an opportunity to cross over more into the service sector and really make the clinician, the physician, more efficient through individualized medicine. Certainly the classical areas of process synthesis, product design, manufacturing are really very much open and of interest for our community. I believe we have a foundation in PSE methodology that George's group contributed in a significant amount, that it makes me very optimistic that these goals can be achieved. Now, I would say the biggest challenge is that George and present company make sure you're still around by 2040 so you can actually benefit from this stuff. So if we deliver it to you, to me and to you after that date, not so optimistic. I will promise, insofar that's under my control. Now, let me just wrap up with a little photo. It's traditional to show a different picture of George. I didn't want to use the same one that's in the program. I thought this was very characteristic of how you were described in this community as someone who's pointed the directions for new ideas to be introduced to PSE, whether it's AI or control synthesis. I'm sure that as you transition to the next phase of your life, you will continue to point areas in which we need to do more work. I'm sure you'll continue to keep prompting Richard to keep working harder so that we get advanced methods into the healthcare area. So with that, I do want to wish you a happy 70th plus one day, and hope that you will continue to optimize, including in your retirement.
Thank you, Rex. So are there any questions for the speaker? We have time for just one question, a quick one. Yes, please.
Yes, Rex. Could you give us some insight into what kind of problems related to control are you envisioning being encountered in this area of manufacturing on demand and essentially personalized medicine?
Well, I think I certainly wouldn't want to minimize the impact of or the need for control. But I believe, as Richard pointed out, I believe we have the tools to actually do it. From my perspective, in the case of for example, flexible dose production, we simply have to find new measurement methods for assuring that the product that we make is correct. So for example, in the drop-on-demand rig, there's an image processor that makes sure that every drop has the same volume. You use ultrasound to make sure you have the same density. So some of the efforts are really on sensing so that you can assure quality. And if it's not, then you reject dose at a time, preferably, rather than batch at a time. I think to my mind, the principal challenges are really measurement. And as I pointed out, and even in the case of the traditional tablet, most of what you measure, you have to use spectroscopic methods to get information. There is no orifice meter for measuring powder flow. For example, we've done work on using X-rays to actually measure particle flow in a process. So sensing, I think, is the big deal. You guys in the control community have it locked down with all the methodology. I think we just have to apply it intelligently.
Well, thank you very much. So our next speaker is Daehee Huang. He obtained his master's in science from Seoul National University. Then he obtained his PhD from MIT in systems biology and bioinformatics, co-supervised jointly by George and Greg Stephanopoulos. His research was very prolific, producing around 14 publications initially. Then he decided to join the Institute for Systems Biology, and 3 years later he moved to Korea, where he assumed a faculty position at Pohang University of Science and Technology. He quickly established a research group that excelled at systems biology, and later in his career he moved to the Department of New Biology in the Daegu Yongbuk Institute of Science and Technology. His current research interests include using high throughput techniques for network. Sorry, I lost my gloss. High throughput technologies, network and biological network modeling, and identification of key pathways in systems biology applications.
Okay, thank you
very
for
much.
kind introduction. Okay, what I'm going to talk about today is systems approaches in biology and medicine, called systems biology and systems medicine respectively. And since I graduated from MIT, been working on the systems biology which sounds far from chemical engineering. I don't belong to chemical engineering department anymore, purely biology now. And yeah, this was me 18 years ago. And when I joined the 2 to 3 months after I joined the lab, George told us that he decided to go to Japan as a CTO of Mitsubishi Chemical. And I was like, wow, I just joined the lab. And when am I going to graduate then? I was worried. And so yesterday I was looking on George's CV, and I found that I participated in most of his bioinformatics and systems biology project undertaken in the lab. And anyway, so it seems like I opened the field in the lab and probably closed the door. The field in the lab. But So on the contrary to my worry, and I could be able to graduate sooner than I expected, and this is photo taken on my defense date, and these are my lab mates, and this is my family. And so after the defense, you're supposed to wait outside until the thesis committee makes decision, right, about regarding my graduation. And I was waiting outside, and George stepped outside, and I was expecting congratulations and etc. to me, but he approached to my wife, and she said congratulations to my wife. So I was graduating while he's congratulating my wife. And he said, yeah, your wife did more work than you for your graduation. That's why my wife really likes George, and she wanted to really participate in the honorable meeting to celebrate his retirement. But unfortunately, my first daughter is now third grade in high school. My wife has to do a lot of things for her. But anyway, so after my graduation, I was getting several offers from Harvard, and I didn't get the offer from Stanford, but Princeton, Yale, and I got an offer from the Leroy Hood in Seattle. And I was talking to George, and which offer should I take? And then he said, you should go to Seattle and work with this guy, Leroy Hood. So yes, I followed his instruction, and I joined this Institute for Systems Biology for my postdoctoral studies. And this is what he thought in the beginning, in early times of systems biology. And you define the problem, and you define the system, and you collect a lot of data, global level data, mRNAs and proteins and etc. And in the beginning, he was talking about developing really quantitative mathematical models, which can be used to predict when you're going to get the disease, and if you treat a particular drug, and when it's going to be better. That kind of really quantitative prediction through these numerical models like ordinary differential equations and partial differential equations, etc. But a little bit later, he realized that there are no kinetic parameters can represent the interactions among the biomolecules, like diffusion parameters and association and dissociation parameters and etc. So we couldn't actually do this quantitative modeling in biological system. So he switched his idea into qualitative modeling here, so which is called network modeling. And here, in network model, you only indicate the interaction among the molecules. Sometimes this is undirected graph, so you don't even know whether this molecule activates this molecule or this molecule activates this molecule. So this is undirected, really qualitative. So in systems medicine approaches, people understand disease. This is the healthy network, properly functioning network. But if you get disease, some of the aspects of these molecules in the network, they will be perturbed. So for example, abundance of this molecule can be increased, or activity through phosphorylation can be increased. So you have the structural change. So you no longer have interaction between these two molecules. So systems medicine approach, they want to reverse this perturbed network into this properly functioning network. Maybe optimization can be involved for that reversion. And this is general procedure in systems biology. This is very similar to systems identification and process modeling, or even problem-solving skill. So you define the problem and you define the system to answer that question. And you perturb the system to gather relevant data on your question. And you generate all types of global data, mRNA data and protein data and phosphorylation data and even metabolite data. After you get this global data, you identify key relevant molecule associated with your question, and after you identify these molecules, you develop a network model which describes the interactions among the molecules selected here. And then by analyzing that model, you generate a hypothesis which can answer your question defined before, and then you experimentally validate this. So since I started my academic career, I've been developing the resources and the tools for systems biology approaches. And for this, I was focusing on this, have been focusing on this 3-step particularly. And in this, once you have global data, sometimes this global data is huge actually. So for proteomic data, sometimes it is beyond 3 terabytes. Anyway, if you have this huge amount of data, you need to have analytical tools which can analyze and visualize, and also you can identify certain molecules associated with your question. And also, how can you actually develop a network model, and how can you analyze this network model to identify certain drug target or diagnostic markers, etc. So these are the two representative tools that I have been developing. And first, when you have a multi-types of global data, how can you actually analyze them? How can you effectively integrate them and to make a conclusion? And also, proteomic data is huge normally, and how can you effectively visualize and integrate them with other types of data? And other tools like how can you, after you have a network model, how can you identify really key, important, essential regulatory structure, substructures in the networks? And this is all about the networks, and how can you identify subnetwork which is closely related to the disease, the certain aspect of disease that you're looking at. And this is like how can you identify temporal evolution of the network along the course of the disease, etc. And as an example, I will talk about this in more detail. Oh, oops, what did I do? Okay. So when I joined the postdoc in the beginning, there were many plant scientists, and they were interested in doing systems biology in plant. And I was looking at the data, but there are not many databases which provide the interactions among the molecules like this in the plant. And the most important thing, so that's how, so before I started any research, I had to collect all the interaction information in the public domain. So we have a separate databases which provide a certain set of interactions. So I had to collect all them all, and they put them in an integrated format so that I can pull them out effectively for a certain system. So this is kind of a pathway model that I defined for the first time in the plant. And you have auxin signaling and ABA. These are the hormones acting in the implant system, and these are their defense, the pathogens, and etc. So, but after you collected all the interactions and the abundance informations after treatment of these hormones and the pathogen, you have now raw data to work with. Then what are the questions you are asking? So I've been talking to these plant scientists at postdoc, and they were interested in interplace between these pathways. Interplace becomes very important because it defines transition from growth to maturation and maturation to senescence, aging. balance between these two pathways can shift from growth to senescence or growth to maturation, etc. So they were interested in interplays among the pathways. So I developed the tools, automatic tools. If you click, then you can identify the really important molecule which govern the interplays among different pathways in plant. So here, so you can just click the data set, and if you add them on, they will be added to the system. And if you click analysis, since this is really complicated data and the essence is divide and conquer algorithm that we learned from PSA, so we perform the clustering and we identify certain cluster which defines interplays between this hormone, salicylic acid hormone, and infection, pathogen infection response. And this cluster also includes a lot of many molecules with a high regulation power. You have many clusters, but they don't have that many molecules with high regulatory power. So I focus on this cluster, and if you look at the gene which belong to this cluster, and you can see they are involved in defense response. So if you click this cluster, if you select this cluster, and if you click the next button, then you will have this kind of a network model. And network model, this is the defense network, and this is a hormone network. And you can see several molecules. They have many interactions with this molecule in this pathway and molecule in this pathway. Those are the guys who can be considered as a candidate molecule which can govern the interplays between these two pathways. So this is one example for this INID system. Oh, I see. And the second example is this part here, how you can identify the important substructure in the network. And the plant have a really strong, the comprehensive network for circadian rhythm. They get this light as an input, and you have a circadian system which generate this pulse, and you have a morning pulse and an evening pulse. And one of my collaborators, he found this GI, Gigantea molecule, which is very important in regulating this core oscillator in circadian system. And he doesn't know what's the regulatory structure. So if he validate which model, identify which model is actually true using experimental technology, it takes forever. So he asked me, what is the regulatory structure? And he can just validate the model that I'm proposing. So I enumerated all the possible structures. These black lines are known interaction, and these red lines are I postulated, the interaction that I postulated. So I generated 5 potential structure. And for each structure, I developed a mathematical model. This is ordinary differential equation. And then, but if you don't know the kinetic parameters, right? So rather than measuring these kinetic parameters using the SPR and all this, I just assumed certain set of the kinetic parameter and then did the simulation, then compared with the experimental data. And there are infinite number of kinetic parameter sets, so I defined the feasible space of kinetic parameters and then did more than 1,000 simulations for each structure. And if you compare your simulation output with the experimental data, you can define the error. This is the shape error. So this is the phase error. Your simulation, in the previous slide, I showed you a simulation here. This is the peak point, and this is the peak point in the experimental data, and you can compare them and then calculate the difference. That's this phase error here. And the shape error is the difference in the width of this path. So wild type wild type, if you project this wild type data into this error space, that will correspond to this origin here. And if you compare all this structure output, the simulation output for this 5 structure, you can see this structure 3 has a highest likelihood of generating the wild type, the oscillatory pattern. So I selected this structure. This is maybe right. And I passed that into my collaborator, and he did the experiment and proved this structure is actually true. And this structure has incoherent feed-forward loop structure, which generates a pulse. So we put them into the paper and published and anything. But anyway, so these are about development of the resources and the tools for analyzing the global data set and etc. And from here, I'm going to talk about the application of these tools. And see, with these tools available, I've been applying these tools into many kinds of refractory disease, rheumatoid arthritis, and type 2 diabetes, and cancers, and neurological disorders, and etc. As an example, probably I can talk about this prion disease. And when you have a network model, the temporal transition of this network model is very important, because sometimes this molecule is cause, and then they will produce the effect. But the effect will also start to regulate the cause. Now, then cause becomes the effect becomes cause. So through this feedback loop, later on, if you look at the snapshot at the later phase of disease, then you don't know which one is cause and which one is effect now. So people want to see how the network evolves over the time. And this prion disease is the disease you have accumulation of a harmful protein called prion. So up to here, you don't have any visible accumulation, so no perturbation in the network. But when you see small amount of accumulation in the brain, you start to see early response. And you have a more accumulation, more perturbation. And you have a lot of accumulation, you have a lot more perturbation of the network. But up to this point, you don't see any clinical symptom. The mouse really look normal, but you have a lot of perturbation in the network, and you still have accumulation too. So if you understand this temporal transition of this network here, you can find early diagnostic markers, and even you can find probably early the drug target, that you want to target the drug target for the treatment. But here in this temporal transition analysis, network analysis, it requires extensive amount of knowledge regarding this disease. Drawing this network, it took about a week for me to draw this initial network here, because I have to study how these individual molecules are actually associated with this clinical pathological features in this disease and etc. So I wasn't happy with that. So I developed another tool which can automatically find the among different parts of the network, different subnetwork in the entire network here. So these molecules in this network, they start to form interaction between these two networks, and then they along the time, and this interaction between these two networks disappear, etc. Here in this network, you don't need the expertise in this knowledge of the disease, but if you input the data, global data, and these kinds of conventional annotation of functional annotation of the genes, then this tool automatically finds the interactions among these different parts of the network. And second application that I want to talk about is the mitochondrial disorder. Here, if you have a defect in mitochondrial DNA, and mitochondria will start to send this SOS signal to nucleus. And if this defect is too severe, then they commit suicide through apoptotic pathways. But if this defect is not that severe, then nuclei respond to that SOS signal to compensate this defect. So they send a lot of firemen to put out these fires here. But if you have this kind of a loop going on in a chronic manner, so defect, SOS signal, and nuclear response, if you have this chronic cycling, then eventually this chronic state actually exceeds a threshold value, then you start to see disease phenotype. So the problem here is this retrograde signaling. If you cut out this retrograde SOS signal here, then you're not going to have a chronic cycling. So as a drug target, many people wanted to know what is the key retrograde signaling pathway in mitochondrial disorder. So that was the question. And for that question, we decided to use this system here, hybrid model. In this hybrid model, you have a mutation in this location here, 32 and 43rd position in mitochondrial genome, this A. If you have a mutation there, A to G mutation, so A becomes G here. And this is a leucine tRNA. It's supposed to fetch leucine amino acid and then deliver that leucine to this protein. But if you have this mutation, this wing, right wing, will be open, and you're not going to have the fetching affinity that much. So you're not going to have the fetching of leucine. So whole entire protein synthesis in mitochondria will be decreased. And mitochondria are supposed to, so you see these kinds of mutation in like 1 to 2% of like a diabetic patient. And we developed a model, so you got the platelet, and platelet doesn't have a nucleus, so it's very easy to isolate this mitochondria from patient. And once you have this system, this is the wild type, you don't have any mutation, and you have, so one cell can have a multiple, more than 300 mitochondria sometimes. So 60% of mitochondria carry the mutation in this condition. Here, 100% of mitochondria, they carry the mutation in this condition. So you have 3 different conditions, wild type, 0% mutation, 60% mutation, and 100% mutation. You generated global data. And of course, this is very complex. So you're getting like 2,000 genes affected by this mutation. So you did clustering. And then you focus on certain cluster, which is supposed to be closely associated with these 2 representative biological processes. So these guys actually include genes indirectly or directly associated with this mutation effect. But this cluster has largest impact on these 2 representative biological processes, and it includes so many regulators. So there is a high chance that this cluster includes the key regulators involved in the retrograde signaling that I talked about in the previous slide. And so you selected this cluster. Now you have to identify the regulator which govern the activities of retrograde signaling. So this cluster includes 500 genes, and I selected a set of transcription factor which regulate the expression of this 500 gene in this cluster. I found out statistically significant, a list of transcription factor with the statistically significant regulatory power for the genes in this cluster. And many of them actually form a protein complex with this guy here. This guy is a well-known protein which regulates a lot of physiology in mitochondria. And some of them actually form the complex with this guy. And among these guys, one molecule, this guy here, actually showed alteration in its abundance caused by this mutation. So after you identify these candidate, drug target candidate, you can actually select this molecule because it shows alteration in its abundance compared to other molecules didn't show any alteration in their abundance. So you generate a hypothesis. So in this case, question was trying to find the key molecule in retrograde signaling, and I defined the system, generated the global data, and I selected this molecule as a key molecule after the network modeling. So based upon this key molecule, I generated this hypothesis. Because of this downregulation of this molecule, you're going to have certain defects in these two representative processes. And my collaborator actually experimentally demonstrated this is actually true. So that's the example of this systems medicine approach. In future, there is currently the frame shift going in this area, systems biology. People have been looking at only mRNA in the beginning. Now people realize that, mRNA only provides partial information regarding the disease pathogenesis. So we should start to other types of dimension. So copy number variation, methylation, mutation, and even proteins and microRNAs. So it's going into now multidimensional genomic analysis. But each different types of data, they have different sizes and different scale. Different size means microRNA, only 500 microRNAs, but this is 20,000 mRNAs. And so they have different size. Also, they have a different scale. The fold changes caused by certain mutation, and these mRNA show like 10 to 20-fold changes sometimes, but this microRNA shows only, as an example, 2 to 3-fold changes. So if you analyze just them using equal weight for these different types of data, then it will be biased toward the larger data set with the large scale. So we have to figure out how we can effectively handle these differences in scale and size and etc. And also the protein data, they provide the phosphorylation information, which other types, they don't have any information regarding the phosphorylation. So there is overlapping information among these different types of data, and also they have distinction in their information. So coverage is different. So we have to effectively handle this complementary nature as well. So this is an example here. If you're using mRNA only, then you cannot actually distinguish these two clusters because mRNA were all upregulated in these two clusters. But if you use the protein now, this protein signature will only upregulated in this cluster here. So you can distinguish these two clusters by using protein data. This is the beauty, complementary nature of this multi-omics analysis. And also, sorry for this Korean. The single cell systems biology now, if you look at this really advanced stage of this colon cancer, they become metastatic. And if you zoom in and then if you look at the tissue, it's composed of many different types of cells here. And if you treat the drug, and some of them will survive and they actually responsible for drug resistance. So people now trying to understand each individual cells rather than this ensemble or average the information by analyzing whole entire tissue. Because the information they collected from entire tissue, they cannot explain the drug resistance among these different patients and different individuals. So they now start to analyze these profiles, the information profiles of individual cells now, and trying to identify key players. For that key players identified, they want to apply the systems biology approaches to understand the key molecules and that should be targeted by drug and etc. So single cell omics technology is going on. Last topic. Okay, so big data analysis. I'll skip this part anyway. So big data can actually provide a lot of hidden relationship among the molecules. And we can develop a network model based upon that hidden relationship in the big data. So you can actually collect all the big data published before, and you can reanalyze them to identify the important relationship hidden in the data. Okay, lastly, happy birthday and happy retirement, George, and thank you. Thank you for being my mentor. And that's it, and I'd like to answer your question.
Very short, quick question for Dehi. I think in the interest of time, we'll keep rolling, and there will be that opportunity during lunch. Thank you very much.
Let me make one comment, so to dispel any misunderstandings. I went to Mitsubishi, and the day he went to work, and he was an extremely hard-working man. That's one of the reasons that he put his name in 14 or 16 papers or something of that sort. The other reason that should be acknowledged is that Greg was there all the time. That made a big difference. Right?
Right.
Now, I can dispel any misconceptions that you might have formed with my magical remote capabilities of directing more efficiently Deihi's work. But
Thank you.
Deihi.
Very good. Our next speaker is Andreas Lininger. He was educated in Austria, getting his diploma in chemical engineering. Then from Vienna University of Technology, 2 years later, a business management education degree. And later on, again, 2 years later, his PhD in chemical engineering from the same institution. George and Andreas met in 1992, and imagine there will be a story about that. 2 years later, Andreas joined MIT, the Laboratory for Intelligent Systems, where he was a principal architect in the development of batch design kit, which we saw showcased before. This was computer-aided systems for pharmaceutical development. In 1997, Andreas joined the Department of Chemical Engineering at the University of Illinois, Chicago. And then in 2010, he joined the Department of Bioengineering in the University of Illinois as well. His work involves system engineering approaches, and today he's going to be talking to us about mathematical models, conversations between minds and machines. So Andreas, thank you.
So it's difficult to follow after so much great ideas, but Dehi set me up for what I would like to do is present to you, George, and to all the other friends that have come, a more philosophical view about modeling and the role of modeling. As I practice it in my new field, this is not a typo, I'm cross-dressing now in neurosurgery. So the overview of my talk is very strategic, which could either be a very transparent talk or a profound one, depending on how you see it. So I would like to introduce a view on what mathematical modeling actually constitutes. I would like to pose the question whether mathematical modeling is practical or meaningful or pays off. And I would like to ask whether it is significant as a research theme. And I would like to base my perspectives on my personal experience by showing you examples so that you can judge with the grain of salt you want to see my recommendations and conclusions and remarks at the end. So to the best of my knowledge, the only book devoted to the philosophy of modeling from a very strategic point of view is Ares's book on mathematical modeling. I got a copy. I got actually almost all the copies of the Dober books. But here he offers a definition that a set of equations corresponding to a physical or biological economical prototype is mathematical modeling. He quotes also a logician, Tarski, that the possible realization in which a valid sentences of a theory T are satisfied constitutes a model. The definition that I like the most is a replacement of a theoryless domain of facts by another for which the theory is known. That definition is really, really striking. And I claim that, for instance, this young man here, Turing, he doesn't look like Cumberbatch, but that's the original. His contribution is not in mathematics or not even in engineering or electrical engineering. I think his contribution is that of proposing a replacement of the decade system into a binary world in which all the decisions that you can make are basically boiling down to and or decisions. So if you transform the arithmetic of regular mathematical operations into the binary world, you can use the power of the binary world to make inferences in the binary world and put it back. That made possible computers. And it was not a mathematical insight, it was a modeling insight. So Kostas Pantelidis showed us a model-based engineering approach. I use a slide that basically covers the same topic, but you have some kind of what Aries called a prototype. My specialty is in unknown processes, so I don't model distillation columns and heat exchangers anymore, but I model processes where we don't know how they work. We formulate, typically in the mind, some kind of theories in another domain. That domain is the domain of mathematics, where I have here a few representatives that I often use. It's linear system, nonlinear systems, control systems. But the bottom line is, as an engineering approximation, the rules of these mathematical systems are kind of well known. Because that's the whole purpose, that we learn from the complexity of this system through transforming it into a system where we know the rules. And then we solve the state equations in this mathematical model and make inferences about the original system. This solving is, in our system environment, often done by machines, where we use computer algorithms to make inferences about the states in the mathematical model, but the meaning actually is not about the solution of a mathematical model, it's about physical predictions in the physical pressure state concentrations. So we can derive insights about the domain, and these insights are not essentially generated in a single pass, but they require a continuous adaptation of what we know about the system. So this feedback is actually more important than the realization of one solution. It is the constant comparison between that what we believe to be represented and what we learn about the system. So that feedback is the essence of modeling. Unfortunately, the formulation of models is not trivial. It's quite expensive. If you just look at these artworks that I have lifted from the book, these models were not generated overnight. You had to think about weeks and months to come at these realizations. So it's very expensive to make these models. And here's a rough back-of-the-envelope calculation about how to generate a model based on my personal experience. So my thesis, I developed a process model for the Corex process, an unknown coal gasification process, which took me about 2 years to just generate the equations. A process flow sheet that I used to teach in senior design takes a good part of the senior design course. I was maybe a little slower than many of you who are more competent in these areas, but it took me basically in the order of 45 hours. An expert may make an Aspen model in a day or even less, but roughly in that magnitude. When we listen to all the great works in algorithmic development and computational efforts with millions of acceleration factors, you can see that depending on what problems you have, where maybe it's solution dependent or algorithm dependent, you may add up 10% of the cost that goes into the model development, into the solution, like the CPU time and the hardware, but the rest is all formulation. Also, Kostas gave us a bottom line that was very similar. He said basically, most of the time I spend formulating and validating the model. So this is not actually zero, but it's so small that PowerPoint decided to make it zero. So most of the cost is in the formulation. Here comes now my relationship with George. George, I understood, asked the following question. Can we use, for the expensive part, machines? He called them intelligent systems. Can we use intelligent system to accelerate, make more accurate, and faster the process of formulating problems? I quote only the papers in the period that I was serving with George at MIT, but these were the papers that made me want to work for him. Modella, we have seen Gabriella's talk about a very visionary paper that basically postulated that the generation of equation-based mathematical models requires a transformation from the physical space of unit operations and stream into mathematical equations. It's basically a language, and the language is here in the Modella modeling language. So it is a language that we need. Intelligent systems, there was Jong Hoon's work here, conceptual processes, so there's the prototype reality and then the transformation into equations. My contribution was to be honored to serve with very outstanding individuals, Ajay Modi and Kike. I didn't find your photos, so I just had your names. But Enrique Saramone, I had the pleasure to serving not only one, but I collaborated with two Stephanopoulos, and not the brother, but the wife. So we were programming Eleni and I together for several years. Shaheen Ali. In this group, what we decided, and thanks Rex for introducing BDK, I think you're the only person who read those papers. But what we basically had, just from a philosophical point of view, we had a bed sheet, a formalized natural language. I would call it a chemist dialect. You couldn't express your feelings, you couldn't say how your work is, but you could charge, you could heat, you could transfer. And this credit for that language goes basically to George. He developed the language and said, Andreas, implement it. What did we implement? We had the operational tasks, the transformation in the composition space of chemicals, and Rex has showed us exactly what is meant here. But we had also the opportunity to schedule and sequence these tasks so that the state transformation would be modeled from the thinking of the chemist and transformed in a computer system, which then in a flow sheet, which featured virtual representation of the material mixtures, the compounds, the process streams, and the units, not actually the equipment, but the units. So BDK was a language that emulated experiments in a virtual laboratory. So what it was is really a model generator. It generated models, and then it solved them also. And it used this language, which has a vocabulary that is not that extensive, actually. You could choose just from these operations. But you then conducted the transformation of your experiment in a virtual environment. So BDK formalized or used the formalized natural language as input. It synthesized equations. It didn't have them. It created them by interpreting whether the material was solid or gaseous. It didn't just execute predefined mathematical modules. And it had a virtual representation of world objects, specifically the chemical compounds, as I called it, the material model. When the tool was taken over, I haven't followed it much. It's still alive as Batch Plus, but it kind of was trimmed back to its essence of the functionality of creating quantitative data. But they used now icons to represent the tasks. They hard-coded the equations. So much of the spirit of which we had the privilege to collaborate was lost in favor of a pragmatism that still prevails the vendors. So if model generation in the 90s was an academic exercise, which as far as the citation is concerned was not that successful, is model generation practical now? So here I go from the limited experience that I have, and I would like to use just examples because there is no time for us to go into content. But I would like to talk about an issue that has to do with the oxygen exchange and blood flow in the aging brain. Aging brain is a personal issue. We are suffering from it, and as I will show later, it's a significant problem. So it's not that we were the only ones who are interested in blood flow. The whole community called neuroscience is devoted to studying the brain, or even more specifically, the neurons. What they made were models that somehow tried to understand what was not yet known. So they made models with control systems, with dynamics, with feedback. But if you don't know how it works, how can you already decide how the model should look like? So I had the advantage, since I'm not from that area, to just know one thing. If I repeat the same approach, I will fail in the same way that the community has failed there. So my choices were do anything but what they did. And what I did is to create a model that appreciated the complexity of the structure. If you look at this, there's a reason why the cerebral vasculature is so intricate and forms such a complex network. So what I proposed is, let me not worry about its function, but let me try to capture its geometrical complexity. So thanks to development that is going on at a very, very fast pace in medical imaging, particularly here, two-photon microscopy, which allows you to basically have an in vivo window into a mouse that is still performing tasks, that is anesthetized or that's running on a wheel, but it is possible to basically create a window, a literal window into the cortex of a mouse and capture the capillaries, the smaller blood vessels, the nuclei of neurons, the nuclei of glial cells, essentially every single cell in that tissue. But with this vectorized image, we cannot yet reason. What we need to do is, and this is why I follow exactly, I think, Deha's talk, we create a network representation of this, which I could summarize as an adjacency matrix. Why? Because the reasoning in this unknown domain will be replaced by reasoning in a domain whose rules we understand very well. So, for instance, these are 4 samples from the somatosensatory cortex of cortical mouse data sets. For me, they're just graphs. And on these graphs, I can perform operations that any person trained in basic statistics can perform, such as the capillary density over the cortical depth, the neuronal density, the particles, the length distribution of these segments, the diameter distribution. Again, we learn about the brain and the cortex by using methods about networks. That's the trick in modeling. But the network itself does not yet reveal its function. So we had to do one more step. We had to breathe some life in that network. And that, from our experience as chemical engineer, was relatively easy. I can fit on one page the equations that we used, or that better said, that we generated. For each node, we have essentially a conservation of mass. There's a detail about blood flow, which I describe next, and the conservation of oxygen between different phases. There's the bound oxygen, there's the dissolved in plasma, then it goes into the tissue. There's some more details, but essentially, it's essentially at an introductory kinetics course, what we used here. What is essential that you cannot eliminate is the fact that blood flows not as a Newtonian or a non-Newtonian fluid. It is a biphasic suspension. It is interesting to see that the community, which I now, since it's not present, may kind of denounce here somewhat, people in fluid mechanics always claim that blood is a non-Newtonian fluid because that's what they're used to modeling. But at the microcirculation, it's not non-Newtonian, it's not turbulent, it's just two phases. So you need to choose the model according to the reality, not according to your expertise. And our expertise is certainly not multi-phase blood flow modeling. But since it appeared in the model as relevant, I had no choice to develop a small, but I would say relatively significant drift flux model of biphasic blood flow. So I looked at the essence of what it's actually doing. There's essentially a velocity of the core that is faster, if you have looked at the cells, than in the boundary layer. And if I simplify that, and I thank that now to Eric Itzty. Eric, I had this intuition about the minimization of the dissipation of the energy, but I would have said, Andreas figured it out somehow. Now I can go to the quotes of the big thinkers that say the system is energy minimizing. This is how blood flow actually occurs. It tries to maximize the blood flow or minimize the dissipative energy in the 2 phases. Now you put in some kinematics and this and that, you will get out the blood flow. This has never been done. I can't wait to go home and implement it. We have done predictions of the microcirculatory blood flow and oxygen extraction. I don't have time to really talk about the brain. I have to stick with the methodology. But I can say proudly that this has been tried for decades. And because the models were always assuming to be continuations of earlier models, people never arrived at anything. We tossed out all of that and said we model every single cell and then build it up again. And we have now a picture that shows us the oxygen saturation, the blood pressure, the RBC velocity, the hematocrit, the Farrois-Lindquist effect, which is that the hematocrit doesn't distribute evenly as a dye would, but that it concentrates in the larger vessels. All these details come from the handful of equations that I showed you for the first time. Also insights that people were never able to infer. When I look at the path through the network, one would assume that in an average capillary, some kind of average mass transfer and average concentration gradients occur, an average pressure. Well, the fact is that each path has a different trajectory, and that the network is rather unevenly distributed. The hematocrit has peaks that reach almost 100%, almost just red blood cells, and no plasma in these vessels. The details, again, I cannot go into, but there's key findings that are not important to me, but important to the neuroscience community by choosing us on the cover of January 2017, which is a flagship of neuroscience, cerebral blood flow and metabolism, where we propose that hemodynamic states are not uniform. And not only do we observe them to be not uniform, but now, with the help of our training, we can say what's the purpose of this kind of randomness. It has the purpose of stabilizing the oxygen concentration in the tissue by using what otherwise would have a controller would have to do. Here it's the space itself and the randomness of the space that creates uniform oxygen perfusion, despite all the variability in the blood supply. Also, we could clarify a question that the pressure drop does not occur in the large arteries, even though they were studied most, because they're most accessible. The pressure drop occurs mostly in the capillaries. So I don't know yet where blood flow is controlled, but I can surmise that if you control the capillary blood supply, it has the largest gain on the blood flow. So I'm close to either having it this way or that way. There's no way that we can stop knowledge gain by using these network models. Now, this was the mouse. Now look at the human. We don't want to talk about mice. We want to talk about the human. But in the human, the two-photon imaging is not available because it requires to take a window in your brain. You need to take the bone off. There's very few volunteers in my lab that would like to do that. So we had to create a model that was equally representative of the system, while at the same time not come directly from an image. So here I'm indebted to the work of Wolfgang Schreiner in Austria, professor of bioinformatics, who had this ingenious observation that if you start in a space with a segment, and then you randomly put another point in that tissue that also wants to be supplied by blood, you could do that by randomly choosing a point and connecting it to that one segment. And then you could say, well, each point that is a so-called terminal node should supply equal amount of blood. Why? Because each tissue has approximately the same metabolic demand. But in order to create a pipeline network, or a tree if you want, that connects those terminal nodes, you want to minimize the effort for the system to grow it. So you minimize the lumen of the tree, or Eric, it minimizes again the shear dissipation. So shear stress-related dissipation, it's analogous. So what you do is you create samples that add one by one another terminal node, and at each step you minimize the lumen of the tree. You have to choose both an integer decision, which segment you add it to, and then you have to minimize the coordinates of that addition, and then you have to compare it to all the other possible trees to find how that structure grows. It's probably the most comprehensive, the most complex algorithms I've ever written. It is recursive, it uses optimization, it has integer decision, and it has to be globally optimal at each step. But let's not talk about the theory, let's talk about the practice. This is an old video. It doesn't contain the thickness because I account for each step. The actual resistance is optimized in such a way that the distribution of each terminal node has exactly the same flow and the minimum tree. So it creates these structures which are total artifacts. There is no blueprint for that tree. It comes out by the principle of optimization. And to show that the power of that algorithm, I have here typed the name of my laboratory for product and process design, written in blood, where each point discharges exactly the same flow. So this is not just some letters here like an L, but each terminal node discharges exactly the same flow. So this, I think, is very close to how nature decides on blood vessels. Here are some graphs that shows how this is done. The main thing I want to show here is this. Look at these trees. This is probably not only correct, but it is aesthetic, it's beautiful, it's appealing. And we have forgotten a little bit in our world the appeal of beauty because we have replaced it with equations. But in the Renaissance, like the Gothic cathedral, before the Gothic cathedrals, and in the Renaissance, the architecture was designed by aesthetics. Leonardo da Vinci didn't compute his bridges, he made them look right. We have forgotten. He studied turbulence, actually, by drawing it. If I can draw it, I can understand it. But judge for yourself. One is the real structure obtained from two-photon imaging. The other one is synthesized by an algorithm. Now, if you look carefully, I think it will not be hard to see that this straight edge here, where I had stopped my domain, is probably the artifact. So that's the human cortex, and this is the mouse cortex as measured by two-photon imaging. But statistically, the topological properties of those two trees cannot be distinguished. They are very similar. So we could extend our experiences from the mouse to the human cortex and answer the question as to the radial and gradial oxygen gradients. These are oxygen maps that show the tissue pressure along blood vessels. And we have published about the system, how we believe it works. But the question is that significant? Is that real research, or is that modeling for the sake of writing papers? Well, knowledge discovery in the aging brain. Rex gave a very, very strong introduction, so I don't have to repeat him. But basically, this is not a waste of time. This is where the future lies for many of the scientific problems of our time. It's the aging brain. It's healthcare related to brain diseases. Here's just some number about dementia. These are $100 billion spent in Europe. That's about $5,500 per person per year on brain-related diseases. 1 in 4 of the senior people, that group I'm slowly approaching to, will have a disability related to the brain. I can live with a limping leg because I fell yesterday, so I'm limping a little bit. But can you imagine that they take your mind away from you? It's probably the worst that we can imagine. Should we work on this problem to keep our minds healthy? You're not thinking doesn't help you. Being famous doesn't help you. All these people had brain diseases. And one statistic that I like, it's not really up here, is by 2015, it's projected that Alzheimer's disease will amount 14 million people. I live in the beautiful state of Illinois. So imagine the whole of Illinois filled with people that have Alzheimer's disease. So one state out of 50, a very important one, completely filled with people whose mind goes down the drain. I have no answers for you today about the aging brain, except for saying that there is a prospect of understanding this as a metabolic disease. These are brand new simulations. They're not published. They may not be true. But what we have seen in the lab, in the two-photon imaging, that leukocytes, which is a special type of blood cells, tend to clog for a short amount of time, 5 to 10 minutes, small blood vessels. They're supposed to do that when you have an inflammation or when there's a cell, a foreign protein that invades, but they are normally not clogging your vessels. But in Alzheimer's mice, there's a tendency of some blood vessels statistically be constantly clogged, then unclogged. So what is the effect of 5% of your blood vessels, of the aging brain, of the Alzheimer's brain, to be clogged? You can see here in red, these are high flows that pervade deep into the tissue. But at 5%, the red goes away like a tequila sunrise, right? It shifts. And at 20%, you have no red at all. It's just blue. That spells bad news for the brain because it's underperfused. There is also an effect on the pressure. You can see here it's low deep in the tissue, while here it's high. This model is probably wrong because the system reacts. It doesn't just wait for the pressure to go up. But that is still significant, Eric, because I believe the brain uses the pressure to control blood flow. So if the pressure is no longer a controlled variable, but is basically taken over by the physics of just going high, you can no longer control blood flow. Now, that cannot be healthy. If an organ that is designed for being dynamically perfused, that suddenly it has no more controllability. That probably has something to do with the neurons losing their function. They live 100 years on their own. It's not a neuronal disease. It's probably a metabolic disease. Now, if there's only one thing that you remember from my talk, that I want to mention it now. Everything I've read about the brain basically says if your perfusion goes down, it's bad for your brain. So you need to keep the perfusion up. And George, thinking doesn't help. Because thinking makes many things go, but not the perfusion. So you need to run, or you need to lift weight, anything that brings your blood pressure up. The brain can help it, but getting more blood. So my little recipe as an engineer is, in the case of a doubt, we don't know for sure what is the truth, but keep your circulation up. Not because you want to stay in shape, but because you don't want to lose your mind. There's two other examples which I briefly mentioned, just a second, which is blood flow in the entire brain, not just in a few blood vessels. Our friends in CFD, they do simulations of one bifurcation. Our lab does simulations over the entire arterial tree by using new techniques like parametric meshing. What do? We want to perform endovascular interventions on the computer and find out whether hemodynamic risk factors are improved before we do the intervention, not after we have done the intervention. We do rational design of gene delivery on the computer first to find out what would be the best way to inject, for instance, enzyme replacement therapies or gene vectors into the central nervous system. And we want to have doctors make the decisions as opposed to engineers. So here the key is not the CFD simulation that I show how the drug moves in the pulsating CSF, but what the key is here that the doctor is enabled to explore his system. So if you want, it is a system that translates the surgeon's intent into input for a CFD computation, which is then launched. The CFD itself is routine. It runs a week on a computer, but that's a different story. It is the intent of what the surgeon wants to do that is necessary. So allow me, after these examples, to come to the conclusions. So modeling accelerates the discovery about complex systems. Modeling is not an academic activity. It's not a mathematical activity. It's essentially the same as saying, I will not learn about the systems. And I showed you that we learn about the aging brain, about drug and gene delivery and systems biology, although there we had a whole talk, so I will not make any comments on this anymore. Formulation is expensive, so that machine generation is both lucrative, meaning reduce the cost, but also effective, because it allows you to think more about your process, opposed to just spending time creating models. This intellectual demand calls for research, maybe systems engineering. I observed that many of us have not entered new areas, but are very comfortable of optimizing things that we already understand very well, which is different from what I proposed here. I want to revisit the question, is modeling effective? And I want to quote here covers that my students have achieved in the last 6 months. Cover of a neuroscience journal. It's very hard to get on the cover. It's our first paper cover. There's nobody in neuroscience who at the same time is an expert in neurosurgery. Other student, same senior author, me, cover of neurosurgery about aquaporins in water transport in the brain. Now forget that and say anesthesia, completely different animal. Here we discuss about how to deliver morphines to the brain on the cover of Anesthesia and Analgesia in December. So it's not my judgment, it's the judgment of my peers that says this modeling has made contributions. My talk was called Conversations about Mind and Machines. So if you believe me or go with me that replacement of a theorless domain of facts by another, then you need to admit that the pure mathematical property exploration yields less about the prototype than a sharpened formulation. What does that mean in plain English? In plain English, it means just studying mathematical properties has zero information content, unless it is tied to the models. And secondly, formulating models is a communication and composition task. So it's a narrative that is important, not just pure facts. I propose a model of modeling. See, it's very easy, and I said it's possible that you think that I'm shallow. But what I wanted to give you an idea is how I talk about modeling now. Whenever I say modeling, I really talk about a language, a foreign language. I speak several languages. English is one of them. I also speak Spanish. German is my mother tongue. But when I think of modeling, it's a language. It can be formal or natural. It has syntax and grammar. And without using syntax, you can't really speak the language. Yes, of course, we need to have math courses. And Dimitri, I'm happy that you're here. Of course, we need to know all the math. And I'm a total math guy. But it is not enough. What we need is a composition where we make a narrative that requires practice. So students need to compose and need to synthesize. I put this model of modeling into practice. I did an anthropological study by designing a course where I said, let me try to teach differently. Let me teach mathematical modeling as a foreign language with reading lessons. Can you read what we see on the screen? Yes, A, B, C, D. No. What does A and B, C stand for? So we interpret these symbols in terms of the mathematical realization, but also in terms of the physical realizations. We read. We translate physical properties in quantitative expressions. We make compositions with multi-physics phenomena, and we interpret the physical behavior. So this course was successful for the undergraduate level. My course evaluations are high, but more important, my colleagues think it's important. So they asked me to make it a graduate level course on advanced reading. So the course has changed the way our bioengineers are trained, because my observation was that after 6 or 7 math courses, we repeated the grammar exercises, but we stagnated in the use of it. So let me stay with this proposal here, that modeling is basically a language problem. And think of it as teaching somebody a foreign language. When you go to industry next time with your module, is it worth learning a new language?
Yes.
This brings me to my conclusions, which it is only due and proper to wish you a happy birthday and to thank you for imbuing us with the courage to do things in a different way. The main way you did that is by just being what you are, a person with a lot of abilities. But what I learned also a lot is, I learned also from your weaknesses, because even though many people of you used godlike names, I saw you more like a man. And I thought that it requires a keen observer. But for me, it was helpful to know that such a brilliant mind and inspiring person is compatible with, a perspective of a humanity. Congratulations.
Thank you, Andreas. So is there a question? So, Ali? George, for how long would you like to?
Thank you very much for a wonderful talk and the humor. I just wonder if you've actually also looked at, aside from oxygen concentration, CO2 concentrations, and pH perhaps, and whether you've applied this outside the brain to another part of the body?
Yeah. This is one insight chases the next model. So this model assumes constant pH. As it is obvious to you, the CO2 redistribution not only has another gas in there, but changes the pH. So as soon as you are happy with one model, you just want to do the next. So it is actually what is needed. We are working now on the pH concentration. I mentioned also that we look at water transport, which is a function of osmolytes, not only of hydrostatic pressure. The key is that modeling is essentially like, as I said, running. You need to constantly move. And as you move, Gabriela showed us that there's weaknesses for us in scheduling. We had Kai-Ying talk about the problems in product design. I have a simple proposal. Just develop the models. Just go in there. I don't intend to be wrong, but most of my models are initially totally wrong. But they're new. So what we need to do is engage in this cycle, and we are actually just right now doing what you suggested, to incorporate pH changes.
I will use a quotation later on in the afternoon from Rutherford Harris and Lisa Penn on the mere notion of the model, which subscribes to the same idea that you have articulated, that but let's wait until we discuss that later on this afternoon.
Thank you very much, Andreas. So we are only 40 minutes behind schedule, which is really good news. So it was asked to return to this room at 1:45, please, so that we can recoup some of that time. Thank you very much. Have a great lunch.